Okay, this is going to be a lecture on tools of molecular genetics. I know that this was in the pre-matriculation material. However, I heard most people didn't look at any of it, so we'll give you this to you again. So the background is, in clinical practice, genetic testing is a major tool in understanding our health, and it's becoming more and more important. It can allow treatment for targeted problems caused by genetic issues. And this is going to be especially important as we go through the cystic fibrosis case. So I want to go through some basic background things. So DNA structure, we probably remember this probably from high school, but there are four bases in DNA. Two are pyrimidines, thymine and cytosine, and purines are adenine and guanine. Adenine and thymine have a double bond. Guanine and cytosine have a triple bond. When we get to chromosomes, which we'll talk more about chromosomes next year, the triple bond GC is actually the more gene-rich area of the genome. So mutation is defined as when there's a change. So if the normal base is supposed to be G that binds with a C, if we change this G to an A, now this is, instead of it being GGA, it's GAA. And sometimes, depending on where it is, this can make a big deal or not. So let's talk about changes in the gene. And this is a more simplistic method, but when I talk to families, I try to find ways for them to understand it. So if we think about a chromosome, this is like an encyclopedia, but I like to think about it as a recipe book. So let's say we have 2 copies of 23 different recipe books, which represent a chromosome. And when we open up this recipe book, the gene is actually a page out of it. So again, we have 2 copies of our autosomes for each one of these recipes. So if the sentence is supposed to say the car was red, let's say there was a second sentence that said, I went to the store. The car was red, I went to the store. What if we changed a letter or a word? So a missense mutation is changing either a word or a letter. So the car was hat, I went to the store. If I asked you what you do, you'd say I got in the car, I went to the store. And if I said what color was the car, and you'd be like, I don't know. But what if it was the car was RDD? You'd probably be, oh, it's probably red. You know, you'd probably guess at that. Nonsense mutations, though. So missense mutations kind of depends on where in the gene it is and how big of a change it is, whether or not that change affects how the protein looks or functions. Nonsense mutations, there was a premature stop codon, which I'm going to show you in the next sentence, in the next slide. The car was red, and instead of that, I went to the store. It's the car, and that's it. So what'd you do? I don't know. I was in the car. Where'd you go? I don't know. What color is the car? I don't know. Insertions of either triplet, like whole words, or just single letters can change the reading frame, and that can cause problems as well as deletions. So this is, no one will ever ask you to be able to memorize this. So please do not try. If you're ever being asked to use this, you will be given this thing. But each set of 3 nucleotides are called a codon. And so the RNA has T and the DNA has T and the RNA goes to U. So if it was AAA, the RNA would be UUU, which a tRNA will bring in the amino acid phenylalanine. You might notice that there are some codes that all these bring in the amino acid leucine. So either UUA, UUG, CUU, so on and so forth. So if I went from CUU to CUC, I would still get a leucine, so who cares? Every protein starts with methionine, AUG. And then here are stop codons, UAA, UAG, UGA. So if tyrosine went from UAC to UAA, I'd go from putting in an amino acid tyrosine to stopping the entire thing. So here we have just some examples of base pair changes. So it's the DNA is TTC, goes to AAG, and puts in the protein the amino acid lysine. But what if we go from TTC to TTT? Well, we still put in lysine. So it doesn't change, and this is silent. It wouldn't even make it onto any report because who cares? But what if it went from TTC to ATC? Now we have a nonsense mutation. We get no protein. Or what if it goes from TTC to TCC? The RNA is AGG, and we get an arginine. Now, lysine, that's a different amino acid, but lysine and arginine are both basic, so it's considered conserved. Whereas if we go from TTC to TGC, we end up with threonine, which is polar. And that's actually non-conserved because this is basic and this is polar. So depending on where it is, this would probably make a bigger change to the protein than this would. So here we have translation, and again, translation is we have our RNA that's made off of our DNA and it's used as a template on the ribosomes. And as I said, our first amino acid that gets brought in is methionine for AUG. Then here comes another amino acid on the end of our tRNA, glycine, and so on and so forth. We're making our nice little protein chain. And then our release factor, here's a stop codon, releases this protein chain. When it's released, oftentimes it goes into the Golgi for folding, and it gets folded into the correct shape. Now, if there's a mutation somewhere at the end of the protein, it may or may not be a big deal, but for instance, in hemoglobin, if it's in the binding site of iron, it could be a really big deal. So here's just another example of what we were going over. Here's our DNA, and what protein that would mean is if we had GCA, we'd end up putting in an alanine, but if we go to CCA, we put in a proline. That's an amino acid difference. However, if we go from AAG to TAG, we get a stop codon, which, as I said, is a nonsense mutation. Nonsense, when there's a stop codon, is always, always, always bad. The other thing that can happen to RNA is we make our piece of RNA. Before the RNA can leave the nucleus, it has to be stabilized. And one of the things that happens is introns are excised. And there are a number of proteins that different introns are excised in different end proteins. So sometimes embryonic have 1, 2, and 3 sliced together, and then the adult has 1, 3, and 4. Now, if there's a problem in the intron axon that prevents this to be properly excised, then that could be, we are fine with whatever this protein is in the embryonic period, but then when we switch over to the more adult period, that's an issue. So same gene, we can use it for different things. The next thing I want to talk about is molecular testing. And so, and the reason I wanted to go over that initial stuff is so we can use that information to understand why we're trying to figure out what things are. So southern blotting, we're looking at DNA. Northern blotting, we're looking at RNA. Western blotting, we're actually looking at protein. And I never even heard of a southwestern blot until I was reading through one of your board questions. It's not used very often, but we use DNA to figure out we separate the protein, then we use DNA to interact with it. Disadvantages of blots is there's no single signal amplification, so we need a ton of DNA. So I have to draw 10 cc's of blood for whether we're doing DNA, RNA, or protein. It's very time consuming, and we're trying to use radioactive probes, but we used to use radioactive isotopes, which are dangerous and we try not to use anymore. Now, if only life was as easy as it was with sickle cell anemia, because every single person with sickle cell anemia, the normal DNA for the hemoglobin is GAG, but in sickle cell it goes to GTG, which instead of putting a glutamic acid in, we put in a valine. And like I said, life is not as simple as it is with sickle cell anemia, but I'm going to use that as an example when we start talking about molecular testing. So in molecular testing, what we do is we cut our DNA with a restriction enzyme, and it cuts between about 4 and 6 base pairs add a palindrome that is the same 5' to 3' and 3' to 5', and we have over 35 restriction enzymes. So we try to pick restriction enzymes that are going to help us like cut out a certain gene region and would help us determine normal versus abnormal for that gene. So let me give you an example. So we have a restriction enzyme that cuts here and here, and then in individual 1, it actually has a third cut. In individual 2, it doesn't. So there's a change in the sequences here. So it cuts this little fragment, and in this individual, we get one large fragment that when we separate, it doesn't migrate as quickly. And then in our individual 1, it actually cuts into 2 smaller fragments that migrate further. We then use a probe that will either attach to this one large fragment or to both of the smaller fragments for when we're looking at. So in southern blotting, we cut our DNA with our restriction enzyme. We separate it. We then move the DNA onto a filter. We add our either radioactive probe or fluorescent probe, and then we can look at it and see where it is. Northern blotting is we have our RNA, so instead of DNA, we look at RNA, we separate it, and then we can look at it again using either radioactive or fluorescent probes. And Western blotting, we use protein. So we're actually, the protein is the end product. So the DNA is on our chromosome, but doesn't make a product. We used to do this all the time for something called Duchenne's muscular dystrophy, which we will be talking about when we get to muscle and mechanics, which is later in the spring. But dystrophin is a very large protein, and it used to be really difficult to actually look for mutations. So we'd actually look at the end product. So we'd draw a ton of blood and see if there's actually dystrophin. So we would use this, separate the protein on a gel and see how far it migrated. This is normal dystrophin. This is a control individual. Becker's has a protein, but smaller. So you can see that this protein, you get a protein, but it's a little bit smaller. But Duchenne's, you don't get any protein. So there's just nothing there. So Becker's has a protein, but it's a little bit smaller than it should be. So I promised you we get back to sickle cell anemia using our agarose gel. And so we use this restriction enzyme because it's the one that works the best, MST. So we cut our 2 fragments, we cut a little piece in the middle of the gene. And in normal hemoglobin, we have a GAG, so it cuts here and here, and it also cuts here. So we get 2 fragments. But in sickle cell, it switches from GAG to GTG. So our MST doesn't cut here. So we just get 1 larger fragment. But in normal hemoglobin, we get 2 smaller fragments. And so here's an individual that has a large fragment and 2 smaller fragments. So they have 1 normal copy of hemoglobin and 1 sickle. And here's the other individual who's also a sickle carrier, a sickle and normal. This child got both copies of the normal hemoglobin from mom and dad. This individual got both of the sickle cell fragments from mom and dad, and this individual got a normal hemoglobin from one parent and the sickle from the other. So this is a way to use Southern to determine in a family, because everybody's the same, it's a lot easier. But again, this is a really difficult method to use. So polymerase chain reaction, or PCR, is a much easier method to look at DNA. And so what we do is we use primers for a specific change in the gene. So with sickle cell, because it's all exactly the same, everyone has the same change in the DNA, we have a primer for normal hemoglobin, but then we also have a primer for the sickle hemoglobin. And so we can amplify that up and determine, and if it amplifies, we get DNA. So we have our template and we have our primers. And again, we can either use normal primer or the abnormal primer to amplify the DNA. So here we have our sequence, and we have our primers, and it makes in a couple cycles, and we put it into hybridization machine. It heats, it cools, and we add our base pairs, and we get a ton of DNA if it works. So we'll have, again, one primer for normal hemoglobin and one for the sickle cell. So actually, this is what we do for newborn screening. So we have the primers for the normal and the primers for the sickle cell. And here's an individual, here's a normal, all we have is normal, so this would be an individual that has normal hemoglobin. The individual in lane 2, all they have is a sickle. The individual in lane 4 has both sickle and normal, so this is a sickle carrier. Again, normal, sickle, sickle carrier. So we can tell both normal and sickle carrier. Now I want to move into the technique of DNA sequencing. And Sanger sequencing is still the gold standard for determining the sequence of DNA. And the way we do this is we have a fragment of DNA, and a lot of people got their PhD doing this. They had this little gene, or that they were going to sequence, and so they do it little fragments by little fragments. So they have their fragment of DNA, and there's 4 different solutions. In one solution, they have our little fragment of DNA, and they add their normal bases, A, T, G, C, and a deoxy T. When the deoxy T is added, the sequence does not continue to add more bases. It just stops there. So in this individual, here's our normal here's the fragment we're trying to sequence. It goes C, A, G, and then it added the deoxy right away. And the second time, this time they added the normal, but the deoxy here, so it was able to keep going. And here it went all the way, normal T, and then deoxy T. So we get a number of different fragments, and we separate these fragments on agarose gel. We do the same thing for C, A, and G. And so what we can do is we can then look and see what these fragments are. So this is just a brief video on this. I'm going to turn on the sound. I'm trying to keep extra sounds off of my computer. So I'm going to turn this on and play a little bit of this video.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
Okay, this stops talking after that, so I'm just going to move on. So that kind of gives you the idea that we can then sequence this. Again, it's incredibly accurate, but you'd see how time-consuming and difficult this is. So one thing I want to talk about is mutation screening. There are over 1,400, unlike sickle cell anemia, in cystic fibrosis, there are over 1,400 different changes in the CFTR gene that can result in the function of the gene not working. In some of these changes, it's only seen in certain families. These include missense, frame shift, splicing, and nonsense mutations. But delta F508, which is a deletion of 3 bases, is the most common in 70% of individuals of Northern European descent. So there are three actually common mutations in both the Ashkenazi Jewish and the Caucasian population, and one of them is the Delta F508. So in the Ashkenazi Jewish population, which is a small inbred population, 94% of individuals of Ashkenazi Jewish descent would be identified using common mutations. So even though the risk before having testing is really high, 1 in 24 individuals of Ashkenazi Jewish descent are a carrier for CF, if they're negative for the testing, their risk drops to 1 in 400, which is actually the lowest. In Caucasians, there's only an 88% detection rate. Their risk before testing is 1 in 25, but it actually is only 1 in 208. That's still a good number, but after the testing. But half, just that 94 to 88, actually in half as far as the detection rate. Hispanics, there's only a 72% detection rate. So even though the risk is like half of that of Caucasians and Ashkenazi Jews, the risk actually doesn't drop all that much. Same with African Americans, it's almost a third risk of Caucasians, but after the testing, the prior testing ones is 65, 1 in 86, which is less than, they have a greater risk than that of individuals of either Caucasian or Ashkenazi Jewish descent, and then Asian Americans, only about 49 or 50%. The risk is super low, but it really doesn't drop all that much looking at common mutations. So I'm just using this as an example of things that we understand well, but when you're doing carrier testing, you need to know every ethnic group has a higher risk of one particular disorder or another. And that's why in genetics, we always want to know what somebody's ethnic background is, because it's important for what we call personalized medicine. So for CF, screening panels are available, which test for most of the common mutations. However, as I said, negative testing does not completely rule out that you have cystic fibrosis or you're a carrier. So mutation screening, the benefits is quick, cheap. Again, we can use PCR. We can, we're getting better and better that we can actually look at hundreds of mutations within that area. However, that only works when we're looking at common mutations in the right ethnic group. Limitations, it doesn't tell you about rarer mutations. And if the test is negative, it's still possible that you carry this. So what we do often with carrier screening is we will test a mother to see if she's a carrier for a number of different things. And if she is a carrier, for instance, for cystic fibrosis, I want to be 100% sure that dad's not a carrier. So I will actually sequence his DNA through either next generation, which we'll talk about later, or Sanger sequencing. So this is actually a USML question that I took from one of these books. A researcher is trying to identify a specific protein in a mixture. He subjects the mixture to gel electrophoresis and transfers to nitrocellulose filters. The filter is incubated with an antibody to the protein. Excessive antibodies washed off and the antibody protein mixture is then incubated with a radioactive protein that labels to the antibody. Autoradiography is performed to detect the presence of the protein. Which of the following technique is being used by the researcher? Northern blotting, southern blotting, southwestern blotting, or western blotting? And the correct answer is western blotting. This is the method of using protein. Southwestern blotting, you're actually separating protein, but you're using DNA and not radio-labeled proteins as to tack onto it. But I honestly have never done north-southwestern blotting. I'm just bringing this up because this was a USMLE question. So that concludes this